In this video, I'll introduce you to the tiny proteins, myosin and actin, that are responsible for helping your skeletal muscle cells contract. I'll review the series of steps required to make this contraction happen, often referred to as the cross-bridge cycle. You ready? Let's do it. Welcome to PhysioFlip. To get a good sense of how tiny proteins in our skeletal muscle cells cause muscle contraction, let's first zoom in onto a small section of the sarcomere, the structure in skeletal muscles that is responsible for generating force. In this sarcomere, we can see a variety of different proteins. Here we have the thick filament, composed mainly of the protein myosin, and the thin filament, composed mainly of the protein actin. Along the thin filament, we also have a couple other proteins, including tropomyosin and troponin. These four proteins together will help control if or how much force this muscle fiber's sarcomere can generate. To generate force in our muscles, myosin has to bind to actin. It has to grab it, pull on it, and shorten the sarcomere. However, myosin normally cannot bind to actin because the protein tropomyosin is blocking access to actin. You can see the binding sites for myosin here on actin being blocked by tropomyosin. That's why I kind of like to think of myosin and actin like two teenagers who want to date. Myosin just wants to go out with actin, but there are these parents, tropomyosin and troponin, who are blocking that, preventing it from happening. But for this muscle to contract, we need myosin to bind to and pull on actin. So let's go through the steps that allow this myosin to bind to actin and eventually pull on it to generate force. First, myosin will bind ATP and break it down or hydrolyze it into ADP and an inorganic phosphate molecule. The energy released from this breakdown of ATP is then used to push the myosin back into position to an energetic state so it's ready to bind to actin. But tropomyosin is still in the way. The parents are still blocking myosin from dating actin. Well, that's where calcium comes in. Calcium can bind to troponin, activating both troponin and tropomyosin, which causes tropomyosin to change shape and expose the binding sites for myosin. That's why releasing calcium from the SR was so important. Without calcium, myosin could never bind to actin and would never generate force. Now with tropomyosin out of the way, myosin will now bind to actin, and by releasing the phosphate, generates the power stroke, which is just a fancy name for saying that myosin actually pulls on actin and generates force. At the end of the power stroke, the ADP molecule is also released, and the myosin will stay attached to actin in a state known as rigor. If we want to generate even more force, we need to just repeat this cycle. That will happen when a new ATP molecule binds to myosin, releasing the myosin from the actin. The ATP is then broken down just like we saw before, with the energy being used to push back the myosin and get it back into the ready and more energized state. If calcium is still present, myosin will bind to actin again and repeat the process to generate even more force. Let's quickly recap the steps of this process, often called the cross-bridge cycle. First, myosin breaks down or hydrolyzes an ATP molecule, using that energy to push the myosin back into a more energetic state and get it ready to bind to actin. Myosin will then bind to actin if calcium has been released from the sarcoplasmic reticulum. Myosin then releases the phosphate group, pulls on the thin filament, and slides it towards the center of the sarcomere, going through the power stroke and generating force. Next, myosin releases the ADP molecule, forming an even tighter bond with actin and entering a state known as rigor, in which the myosin has pulled on actin, is generating force, but is still stuck to the thin filament. To release myosin, either to help the muscle relax or to get myosin ready to start another cycle to generate even more force, myosin will bind ATP, releasing actin, and allowing us to start this process all over again. That's a lot of steps, right? But here's the result. Myosin will pull on actin and shorten the sarcomere to generate force. By the way, you might notice that neither actin or myosin filaments are actually getting shorter. All that happens is that actin slides over myosin, being pulled towards the center of the sarcomere, shortening the sarcomere to generate force. That's why this process is often called the sliding filament theory of muscle contraction, since the thick and thin filaments seem to slide over one another.